Olá a todos, sejam todos bem-vindos e bem-vindas. Começamos mais um webinário internacional e temos hoje um convidado mais do que ilustre, altamente, profundamente conhecido, e por conta do idioma, nós vamos utilizar então o inglês para nos comunicarmos. So, thank you so much for everyone to be here. Today you have a very important, outstanding, and it's an honor to be today with the Professor Ulo Langel. Professor Ulo Langel will talk about tools for new pharmacology and cell-penetrating peptides and oligonucleotide delivery. He's from the Department of Biochemistry and Biophysics in Stockholm University, Institute of Technology, Tartu University as well. And on behalf of the Graduate and Research Office, I would like to thank you, Professor Ulo Langel, to dedicate your time for your special research, and also today for being here, dedicating your time to share your knowledge. And there's a lot of people from our university in over the country that wants to listen to you, and that wants to get advices from you, and that wants to know the upcoming news or the new information about the pharmacology. But also, I would like to thank you, Professor Miran Hayashi, who is a very good partner of the graduate studies and the graduate and research studies. And she's been working with us and collaborating and cooperating to organize a lot of webinars. And Professor Miran Hayashi, she was born and educated in Brazil, and she's currently Associated Professor of Pharmacology at the Federal University of São Paulo, and head of the Molecular Pharmacology Lab, and Vice Chief of the Discipline in Cellular Pharmacology at the Department of Pharmacology of the Escola Paulista de Medicina. And she's extensively involved in medical and biomedical education, and is also active in multiple research endeavors, which primarily and focused on the characterization of the association between oligopeptides and oligopeptidiasis and mental disorders. Her works broadly aim to characterize the underlying causes of mental disorders, notably schizophrenia and bipolar disorders, and pursuing the neurodevelopmental hypothesis and the importance of oligopeptidase and inflammation to explain the dual influence of genetic background and environment for susceptibility to mental disorders. This body of work has provided up to now a platform for identifying novel molecular biomarkers for diagnosis and potential new targets to treat and possibly prevent mental disorders. So thank you so much, Professor Miran Hayashi. I'll let you to introduce a little bit better Professor Ulo Langel. It's a great honor for the UniFESP to receive both of you today with us. Thank you so much.
Thank you, Camilo. Thank you also for Dr. Ulo for accepting to give us this webinar. So I would like to present Dr. Langel, that is a professor of the Department of Biochemistry and Biophysics in Stockholm University, and at the Institute of Technology at Tartu University. Professor Langel graduated from Tartu University in Estonia and bioorganic chemistry in 1974. He has received his PhD degree twice in 1980 from Tartu University and also in 1993 at Stockholm University. His professional experience includes career at Tartu University, junior research fellow to associate professor, visiting professor, and professor up to present. He worked at the Scripps Research Institute at La Jolla in California, USA as associate professor until present, and at the Stockholm University as associate professor and chairman at the present. He's an honorary professor at Ljubljana University, I'm sorry for the wrong accent, in Slovenia, and he's the chairman in the board of small company like Peptide in Stockholm and CPEP in Estonia, and member also of the board of Orexo at Sweden. In 2013, he was elected a member of Academia Europaea, and in 2015, a foreign member of Estonian Academy of Science. Professor Ulo Langel is reference in the area of cell-penetrating peptides, and I'm pretty sure that we all are going to learn a lot with him. So thank you again for accepting this invitation. The stage is yours.
Thank you for this kind introduction. I'm happy to present for your university. This is my first visit to Brazil, and I hope it will not be the last. Maybe I can even make a travel sometimes in 5 years from now or so.
Yeah, sure. Give yourself invited.
I will talk about cell-penetrating peptides, and I will start with introduction to the field, and then I will show you our work on oligonucleotide delivery, which I am working basically today, but some side products are always available, so I work with many projects in parallel. I call it the tools for new pharmacology because it explores the new pharmaceutical area, intracellular interactions. It's not only limited with oligonucleotide regulation and gene therapy. It's also involving the, I can say, all intracellular interactions, protein-protein interactions, oligonucleotide interactions with proteins, other regulators, and if you think about it, and you probably know that, this field of or area of intracellular interactions is very seldom today available for drug developments. So I hope this will change in future, and that's why I call it new pharmacology. I have also worked with peptides for many years, and I hope very much that this cell-penetrating peptide field will help us to develop more peptides to become the drugs, because they are not very popular in pharmacology, except some exceptions. They are degradable, and they are not under 1,000 Dalton molecules. So the dogma in pharmacology very often says that peptides are not good for drug development, but this thinking will probably be changed, I think. Now I am involved in Stockholm University and at Tartu University in Estonia, where I have small groups. But as you can see here on this slide, I also have a lot of collaboration. I only took few of them out of the context. So you can see here even Professor Emerson de Silva in Sao Paulo, who we have recently published a common article, and we are continuing the collaboration now. Miriam is involved in our new CPP book, and I hope this can be another collaboration. So let's see how these things will go. Maybe it will be more Brazil collaboration. But I have to mention that I can see more and more activities in this field in Brazil in recent years. So why not? And I want to point out two recent books on the area. This one is a monograph which I wrote on CPPs 2019. If you have any questions on CPPs, I think you find everything in this book. And in November now, this year, the Methods in Molecular Biology book will be published, where I'm an editor and where I have these two Brazilian authors also involved. I want to ask, do you hear me? Because I only hear the silence in my earplugs. Do you hear me?
Yes, we can hear you. It's
okay.
Can
you warn me somehow if you don't?
Yes, sure.
It's a strange feeling. I'm talking to empty space.
Yeah, sorry about that. I can understand you.
No, don't be sorry for that.
But
okay.
I will tell to the audience that if you guys want to make questions, just fill up the Q&A, and then I can interrupt too if it's necessary. Okay?
Yeah, that's possible. Okay. First of all, what is a cell-penetrating peptide, or CPP? It is not easy to say. It's quite diffuse class of short peptides. I wrote here 4 to 30. There is also a 2 amino acid long peptide available in the list, and upper limit is as high as you define a border between a peptide and a protein. So there are all types of or lengths of peptides available, which are cell-penetrating peptides, which are defined with these points here. They enter the cells, they translocate into the cells through a cell membrane somehow, with different mechanisms, which I will a bit discuss with you later. And the most important feature maybe is that they deliver into the cell interior the cargo, which are attached covalently or non-covalently. And I think this is the most important feature also, that they are bioactive, these cargoes. So they don't lose their activity during this process. So that's the most important feature maybe, that bioactivity is available and you can connect different cargoes. And then usually people say that they are non-toxic and efficient and harmless, which several experiments show. And sometimes these peptides have their own activity, intrinsic activity, which is nicely used today for mimicry of protein-protein interactions, which I will talk about later. So as you can see here, there is no strict definition. It includes a very, very diffuse class of short peptides with very, very diffuse properties, biological properties, physical, chemical properties. So that's why it's so difficult to say what the CPP is. Okay. And in this table, I have summarized some of CPPs which are available today. I think this all started in 1994 with the publishing of penetratin by a French group led by Alain Perchian in Paris. Before 1994, patented their discovery of this peptide. They called it first P. antennapedia, as you might recall. Now it's called penetratin, and it's a 16 amino acid long peptide, which has been shown in many experiments to do what the CPPs do, as I showed on previous slide. Then 97, the shorter fragment of HIV TAT protein appeared, also from French group in Montpellier, Bernard Lebleu's group. It is called usually TAT. It is 12 amino acid long peptide, but the TAT protein, it's derived from TAT protein, and TAT protein has several sequences of CPPs inside of them. Then approximately the same time, we patented it 97 and published 98, our transportan came out, and this was connected to my studies of neuropeptide galanin, and making the long story short, it is a chimeric peptide between N-terminus of galanin and mastoperon. It's a wasp venom toxin. And then you can see a lot of famous ones in this table, but I will comment that later. But if you look at what are these peptides? How are they designed? How are they introduced? By what properties? Then on the right side here, you can see that most 80% of them are cationic, but a huge amount is also anionic, huge amount, 5%. Still, it is not only the cationic peptides. Then you can see that a lot of them are amphiphilic and a lot of them are hydrophobic. And you can also see the most important thing in this figure is that these properties more or less can overlap. So every CPP usually involves a different amount of these properties. They are cationic, they can be also anionic sometimes, or at the same time, and they can be amphiphilic and hydrophobic, and they can have elements of these properties in each sequence. And that makes also this class of peptides very diverse. And to classify it is quite difficult. On the left side here, you can see what was done at the beginning. And this is that they are derived from proteins. Both penetratin and TAT peptides were derived from larger protein, which had a biological function. And when it was discovered that a short peptide sequence in these proteins was responsible for the cell penetration of a whole protein. And then these proteins like antennapedia and engrailed and others, what you have heard from today, are so-called shuttling proteins, which shuttled between the cells. And this was in 90s, at the beginning of 90s, it was actually first 87 even, they were discovered with shuttling proteins. And this was a breakthrough in this field, where it was a dogma that nothing can be taken into the cells if it is large and not hydrophobic. And that was very difficult to break this dogma. But I could feel on my own research, it was quite difficult to publish something because the regular answer from the editor was, and referees was that I can't publish it because everybody knows that the peptides and proteins don't enter the cells. But we couldn't do anything because our experiments showed that. And the first source of these proteins was derived from proteins and peptides, which are available in nature. And the first method to find these peptides was so-called educated guess, looking at the peptide and finding a cationic region in this peptide was usually used as a first information to prepare a short CPP, and it worked sometimes.
Dr.
And then,
sorry for interrupting you, but how do you fit proline-rich peptides in this?
Sorry?
Proline-rich peptides. How do you fit in those classifications?
I don't fit in here anything because that was the first possibility to do that, to just select the cationic sequences. The proline things came later, and they are also available in some proteins, but this was not the first beginning of this. And then in parallel to that, I put the designed CPPs, which is a case for our transportant, for example. We by chance put together galanin and mastoperon and ended up with CPP. And some other groups did the same with other peptides, and that was the beginning how they were found. Then you can divide these peptides by physicochemical properties or structural properties. You can classify it. You can say that this type of or one class of peptide is predicted and our class is random. Then you can see the linear CPPs, you can see cyclic CPPs, another possibility. Then you can divide them to protein mimics or cargo delivery peptides, non-specific targeted. By mechanism, you can divide them direct penetration or endocytosis enhancers, and that was long time basis of the classification of them, but this is showing you actually how diverse these possibilities are to classify them. And these 8 possibilities probably are not the only ones. You can find maybe hundreds of such properties which you can use for classification, and any one of them is good. So to say what class of CPPs you are working with is not easy at all, I think, because of diversity of them. Now I come to the problems that I see are the most important in the field of CPP studies. And that's the same poor understanding of mechanisms and why they are going to different places in the cell or even in vivo. And then we should talk about targeting immediately, because if we talk about biodistribution when, of course, we are interested in a peptides which we can take into certain places with certain activity. For example, in development of cancer therapies, you want to have your drugs very nicely targeted the cancer tissue, for example. And another issue here today is that we have very good tools, very efficient tools. We understand a lot about the CPPs in case if they work on live cells, but we have much less understanding for in vivo applications, everything, how to deliver them in vivo using targeting, how to maintain them in non-degradable form, how to get the cargo to these peptides so that you can protect it from degradation as well. And you can name actually maybe hundreds of such problems, but I think these are the most important ones. And if you look at this, if we start from mechanisms and targets, then it's a very, very simplified scheme of a cell and the CPPs, which are these small crooks here. And what you can see is that you can use different cargos in CPPs. You can actually, from short peptides and small molecules and oligonucleotides, you can even use it as a cargo, antibodies or large plasmids, and it seems there is no limit in size, and that's a good news if you want to get into cells, the bioactive molecules, when you see that CPPs can deliver them all. And then another thing, of course, what I didn't mention yet is not only the cell membrane, but in general, all the biobarriers, like endothelial barriers, like blood-brain barrier, and others. Even there, we don't know everything yet, but even there, we have a lot of progress. When commenting this mechanism picture here is using the cell membrane as a target for penetration. And you can see here that there are two basic mechanisms. One of them is described briefly here, and this is the direct penetration, so-called direct penetration of CPPs into a cell, where they can find intracellular targets, and they can find intracellular protein-protein interactions, or whatever interactions they are designed for, like oligonucleotides and proteins or whatever. And this is believed to happen in a way that they interact with plasma membrane, and when they, in this interaction course, find them on the other side of a protein, of a cell membrane. And then there is this classical receptor, cell surface receptor interaction with cell-penetrating peptides, where these receptors are defined with different transmembrane proteins, like 7 transmembrane proteins, like proteoglycans, like ion channels even, and some unknown proteins today. We have, for example, found scavenger receptors in this procedure, which is sometimes important in cellular delivery. Now, it seems, it's not of course so simple, but it seems that direct penetration is a clear entering into the cells. But of course, it is probably more complicated at interactions with both membrane components and then intracellular components are available here. Then if we look at the endosomal escape, we know a lot about endocytosis. We know that when the receptor binds the ligand, and this is a CPP in this case, it will be internalized and it will be found in the endosomes. Now, in the case of CPPs, which are connecting different cargoes, it means that a lot of cargo is found in the endosomes. And actually, this is not what you want. What you want is that they will exit the endosomes and find different intercellular sites like mitochondria or nucleus for their work, for their interactions. And what I still believe is that the CPPs are good to escape from the endosomes, exit the endosomes. We are not very sure about the mechanism, but if we influence the endosomes with classical compounds for endosomal escape, then we will get better endosomal escape as well. So this slide explains everything, what you can do with them, and these mechanisms, I believe, can exist in parallel. The direct translocation, endosomal uptake, and this probably involves many additional processes like pore formation here in a direct translocation, more or less. Antimicrobial peptides are similar with a pore formation, and sometimes, depending on the concentrations, can do the same as CPPs. And I believe that these two mechanisms and the involvement of different receptors can be in parallel. The time will show if it is correct. Here you can see a few examples of different endocytosis mechanisms which have been shown for the CPP uptake by this mechanism, and endosomal uptake, endocytotic uptake is usually more likely, it has been shown, for the cargoes, so bigger cargoes which are connected to CPPs. If the cargo is small and CPP itself sometimes is using only this direct translocation, but also it is a mixed procedure probably. And many types, not many types, several types of endocytosis have been shown, and different receptors have been shown to be involved. Now, I wanted to give you this slide, discuss it a bit, where I wanted to show that CPPs or the cargos connected to them, if we want to design them this way, can interact with signaling inside the cell, or they can even initiate the signaling in the cell. And also this is possible by two different mechanisms, endosomal uptake and direct uptake, which I will demonstrate you a bit later, and to find the intracellular target so that we can influence their signaling. The CPPs can be used for that, and by influencing the signaling, we can influence cellular response. Amazing. If it works, and if we can interact something which is caused by a receptor ligand interaction, if we can do that without involving the receptors, then it's an amazing new therapy possibility. And I will show you that this is possible. And if this small one here is a CPP with a signaling interference, and this big one is a protein, this illustration shows that if we find the CPP sequence, which is designed from such interaction site, then it can bind to the protein and influence the signaling. And this is something which possibly opens all protein-protein interactions and their regulation. The same figure here. I more than 20 years ago had the first examples of such mimicry, and I called them cell-penetrating protein mimics. And this means that the integration of protein mimicry with short CPPs should be possible. And again, I repeat here, if we have a possibility to design a short CPP with mimicry of protein somehow, then it interacts with, here in this example, interacts with a dimeric peptide and interrupts this interaction, for example. It's one possibility. There are more. So we have an extremely important tool for future cell-penetrating protein mimics. And I wanted to say one more thing. This is important because there are several small molecules available doing the same, but they have been found by screening large amounts of compounds of small molecules. And it is possible to find these molecules that way also. But if this is a protein-derived short peptide, then it's similar to oligonucleotide interaction with other oligonucleotide. But here it's a protein-protein interaction. And this is something which is also available today, to design a short piece of a protein which interacts with some other protein. And I go on. I start from the CPP part in this, how to design a CPP today. And I start from our prediction. I started this 23 years ago or so in my group. I don't want you to read this. I want to tell you that this method used different physicochemical variables for 87 amino acids and calculated a certain number, hydrophobicity, steric hindrance, charge, and other increments for each amino acid. So if you 87 amino acids they took, so there are 20 coded amino acids and others are non-natural amino acids, and you can calculate for each one of them, you can calculate the variables. And then you, from that, as done by Svante Wold's group in already long time ago, 87, you can calculate the summary for each peptide with zeta values. I hope this makes sense for you. It's a QSAR approach, which is a very nice one. We were able to predict millions of CPPs, if not billions, based on this amino acid sequence. And it is a bulk property of CPPs, meaning that you see what kind of amino acids you have, and you can calculate with zeta values if it falls into these areas when it's a CPP. And we found CPPs in many proteins. We just analyzed the human receptor proteins, and we found already millions of sequences. And if you generate a random sequence, you can also do that. And here is a patent description and publication on this, so you can look after yourself.
Dr. Langel.
Yes.
A question at this moment. For predicted CPPs, is this only the primary sequence who matters, or because like a Tartu sequences of polyarginine or polylysine, it didn't work that well, right? It
Yes.
was a problem of a 3D, or it's just a question of a primary sequence? What do you think?
It is so that none of these predicting algorithms work 100%. Our prediction works 80%, which is pretty good, but it's not 100%. So you can make a mistake very easily. So I can't answer your question actually. Nowadays there are around 20 different algorithms available, machine learning and neuronal networks and whatever you can find. The problem is that you can't always check all of them. So there is not even available any good comparison between these 20 algorithms yet. I hope it will come, but you know, for each peptide you have to make it, you have to maybe fluorescently label it, you have to test its biological activity. And each time for each peptide, it's a lot of work. So you predict, but these millions of CPPs, you don't know if they are really always working.
Okay, thank you.
And this is a bulk property. So this means that it doesn't matter for this algorithm if these amino acids are in a certain sequence, which sounds wild. But in several cases, like the penetratin, from the first beginning, it was stated that the CPP properties do not depend on a sequence of these amino acids. Absolutely crazy, right? But it's not always so. And I bring you only one example. We took around 800 human G protein coupled receptors, and we run the protein sequence with our prediction. And here is a plot of position in this sequence and the number of predicted CPPs in a sequence of this protein. And if we put them under each other in a percentage scale, then you can see that there is this intracellular loop 3 and intracellular C-terminus, and also this loop 1, which contain most of the CPP sequences, which gave us an idea that we could find here in intracellular loop 3 by prediction the CPPs which have the protein mimicry properties, because this intracellular loop 3 was known for involving most of the sequences which were by that time known for interaction with G proteins and also with C-terminal part. And this we tried with several protein or G protein coupled receptors, hoping that if we are able to find the mimicry site here, and it is a CPP, then we can send it into a cell and it should find the G protein and activate this. And we found them several times. So we found this mimicking peptide from intracellular loop 3, which had the CPP properties, and we sent it to a cell. It found the G protein and starting a signaling cascade. So we have what I promised you at the beginning. We have a protein mimic with CPP properties, which independently from the receptor can activate the G protein system. And I have several examples. I brought only few examples. Here is this GABA B1 receptor interaction with protein 14-free. And what we did, we found a sequence by prediction which interrupted this interaction. 14-free. It was uptaken by the cells and it mimicked this interaction. Here is a short table of several such interactions, which you can find in literature summarized, where we can do the same with other proteins. Now I go to the oligonucleotide part, where we usually use the antisense technology in splice correction model, which is Richard Kale's lab's work, where the cell line with HeLa with a specific sequence, aberrant sequence of encoding or not encoding, actually look 705. If you send an antisense sequence here into the cells, which expresses this aberrant version, then we have a positive readout, because this is taken away in splicing and luciferase will be expressed, and you can see the readout of luciferase fluorescence. This is what we use very often. We have an antisense short oligonucleotide for that, and we couple it to the CPP, and we will see if a peptide is starting to express the correct luciferase or not. Then siRNA delivery for knockdown. So it's a tool for gene therapy in future. And then we usually use plasmid delivery with CPPs very often. So we have covered most of them. Now we have working also with miRNA and other oligonucleotides if interested. And here is a summary where do we act. So we have to get siRNA to a cytosol. We have to get the plasmid into the nucleus, and sometimes we are interested also to work with mitochondria, for example, take tools for therapy into these organelles, and then there are available other sites of interaction. And I introduce you the peptides, which are our transporter analogs, which has a sterile modification on the N-terminus. And here is one of the first, peptide 6, which also had the chloroquine analogs in side chain with lysine, which we added to enhance the endosomal escape. And this is the structure, usually sterile or fatty acid here, a linker, and the amino group, usually in a lysine side chain to connect different cargos. And we can connect covalently the oligonucleotides here, but we also, which I prefer today in my group, is a non-covalent complex formation between the CPP and oligonucleotide. And this peptide was one of the first which was able to deliver oligonucleotides into the cell. You can see electron microscopy of it, and their size was around 100 nanometers, sometimes smaller, sometimes bigger. So these peptides can easily make analogs of them and make them better. Here is an example of siRNA knockdown of luciferase in liver. You can see in time you can downregulate this protein expression, and here is same downregulation in cells. So you can very easily knock down, not knock out, but knock down the targeted genes. Another example here is a tumor targeting, where we use another peptide 14 here, which is connected to PEG to get a better degradation resistance. And here is a cleavage site, this green one, which is cleaved by MMP2. So the idea is that these form the micelles, which are including the oligonucleotide, plasmid, or whatever you are interested in. And then only if it gets close to a cancer cells, tumors, the MMP2, matrix metalloprotease 2, is taking away this PEG sequence, and the CPP will be released for interaction with a cancer cell, and only cancer cells will be able to translocate it. And with this, we have reporter gene induction after transfection of plasmids in tumors, and we also are able to downregulate the tumor growth and survival rate here, the green one, with this type of approach. I will not go into this. We had a very interesting article, which you can find if you're interested, where our technology for plasmid transfection by Peptide was used to confirm the participants of neuroendocrine tumors. And I'm pretty proud that it worked very well for this group to show that by siRNA formation you can knock down functional proteins in this cancer. We have also worked with blood-brain barrier. We have this delivery vector which contains a homing sequence to the brain, angiotensin, for example, or G-HOPE, which was found in screening of libraries, and CPP, and it is possible to get into the brain. The tumor area is here, and we have a slight labeling of a tumor here. So it's a glioma homing peptide 2 for brain tumor, glioma labeling. Also pretty interesting. From RNA sequencing, we showed a very interesting thing that in mechanism, the peptide 14 oligo complexes involved in their uptake process autophagy. This was also shown with small molecule ligands. I will not stop there, and I don't want to comment this in details, but autophagy is involved in very interestingly very many cellular processes, like CPPs are, especially if they are connected to cargo. And we showed here that autophagy is an important mechanism for our particle uptake. Then maybe you noticed that different non-organic, anorganic, inorganic molecules have been added to transfection reagents, and we tried also several ones. Graphene oxide was added to a CPP plasmid complex here. We don't know how it looks like, but maybe this or this, no idea. But graphene oxide 10 or 20-fold activates this process, which is extremely interesting. How do they look like? We don't actually know. This molecular dynamics in water shows that maybe siRNA connects several molecules of CPPs. Maybe it's like this, or maybe it's like this. We have some data saying that, but we are far from saying what happens exactly. Exactly what this model shows, that there is a equilibrium between the particles and components of it, and all of them can have different uptake routes. So it's a nightmare to solve this kinetic equilibrium system. It's complicated, that's for sure. And thank you for your attention.
Thank you, Dr. Langel. It's a very interesting, amazing presentation. Thank you very much. I will start reading some of the questions. I have a couple of questions in Q&A. Tara, you want to say something? Is this okay?
Yeah.
Okay, thank you. So there's one question that I think that you have already answered, by partially at least. So as you might know, there is a couple of groups here in Brazil working with rattlesnake toxin, named crotamine. So this person seems to have worked with crotamine before, and he wants to know if there is any other CPP showing specificity for proliferating cells, and with
showing what? Say it again.
Specificity or affinity for proliferating cells that could be applied for nanocarrier for oncotherapeutics.
I don't know exactly which ones, but I'm sure there are several. I never studied this problem before, so I will not answer now.
Yeah, and I think that you have shown some of those CPPs with affinity for glioma, and also those for metalloproteinases.
There is a lot of homing peptides available and a lot of ligands for cancer cells, of course. I think hundreds. And homing peptides are also discovered in screening of libraries, so there are more and more.
Okay, but do you know if there is some of them that have already been used in clinics that is being applied for treatment?
There is no CPP in clinics yet. There are several clinical trials. And there is a French company working with cancer. There is an English British company working with Duchenne muscular dystrophy, and there are several others. And actually, nowadays, recently, like 5-6 different big pharmas have turned to me with the same question, and I'm trying to help them. But, you know, the pharmacy doesn't share what they have and what they don't very much. Yeah. So I'm sure there will be the drugs available, but I'm not sure when.
Yeah, I can understand. So for the audience who does want to make questions for Dr. Langel, please just write down in the Q&A. And so I will follow the question since you are the reference in CPP studies, as I said before, and you have more than 400 publications in this area. Do you think that CPPs can be considered CPP only when it works as a small peptide? I mean, because most of the time they are part of big proteins, right? But if you excise them or just synthesize them chemically, sometimes they just lose their ability of targeting specific cells. So what is the right definition of CPPs?
Well, I gave you the definition at the beginning, and this was very diffuse, but that's what you can do. There are several CPPs nowadays which are resistant to degradation, and our peptides, for example, are also resistant because they form the complexes which protect them for degradation. But there are many approaches like stapled peptides, cyclic peptides, which are resistant for degradation. As I said, what we don't know for CPPs is how we can use them efficiently in vivo. That's the problem. And that's what everyone works with.
Yeah, sure. So I do have a question for you regarding those CPPs present in GPCRs, in big proteins that are immersed.
Do you have a bird singing?
No, GPCRs, CPP.
No. I hear the bird singing. Am I crazy? Or is there a bird?
Oh, there's birds. Yeah, actually, there's birds in São Paulo City. Yeah.
Okay, it's good to have them in a seminar.
Yeah, we have a couple of birds in my window.
Okay. I'm not crazy.
Yeah, no, you're not. And I promise you, I'm in the lab. I'm in Delta area, but even though there is some birds.
Okay.
It's incredible, but yeah, they can survive to big towns like São Paulo.
Okay.
So, my scientific question is regarding those CPPs from GPCRs. So big proteins with three-dimensional complex structure. Most of those CPPs that I suppose should be just present high content of basic amino acid residues. Do they keep this CPP ability or characteristics even if you just cut off from those big proteins? I mean, do they need the three-dimensional interaction of other loops?
Well,
of the
the short peptides don't have usually any structure in water, but what happens even if a protein interacts with G protein is an induced fit. So I believe if we get a good mimic, when it has an induced fit with a G protein, for example. I believe it, but it hasn't been shown.
Yeah, I agree with you, but it's very complicated to show it, right?
Oh, yes.
So there is another question. It's from Joanna. She's my PhD student. She says, thank you for your presentation. And she has a question regarding the clinical trials involving CPPs. Do you know which types of therapies they are being applied for now, besides those you have already mentioned?
Well, there are several mimics, protein mimics. There are several oligonucleotides. There is vaccination with CPP participation. There are protein delivery approaches. If you look further in my book, then everything is available in detail. I don't remember all of them, but almost all classes of compounds are available. And you have to consider also that some of these clinical trials have been stopped at different levels, and some of them are still ongoing. And very few are in phase 3 clinical trials. So it goes very slowly.
Yeah, I'm very anxious to see your new book, your new release. So we'll check for that. I have a question for your collaborator, Emerson Rodrigo da Silva. So he's asking if there are some trials on the use of CPPs for designing peptide vaccines.
Again?
He's asking about CPPs being designed for peptide vaccines. So he's asking about vaccination.
Yeah, there are several such peptides available. If you want me to look it up, I don't know by heart. But we are also trying to enter this field with our PepFacts or NICFacts, actually.
Okay, thank you. There is also another question from Professor Patricia Hiskill. She's a colleague of mine from pharmacology department. She's saying nice talk, and she's asking, what do you think about using a TAT sequence to deliver peptides inside the cells? Which kind of delivering peptides inside the cells do you think could be the less toxic to be used by human in the future?
Well, if you are interested in cellular delivery, then I think it can take in all small peptides. Actually, it depends on the peptide cargo also. If it is strongly charged, negatively or positively, it can interact with CPP. And then we don't know exactly. It can be aggregations and toxicity due to that. So to predict the toxicity for peptides is a bit difficult today. I don't know about good works in this field. But if you want to deliver peptides into the cell, then you have to ask the question, how do you conjugate it to the peptide? If you make a fusion of TAT with a peptide, probably it will work, but we usually recommend a connection, chemical connection between TAT and cargo peptide through the disulfide bond, which probably degrades when in a cytosol, and you get a free peptide instead of longer peptide. And it sometimes helps if you have a free peptide.
Yeah, and if you allow me to add to this. I think that sometimes you have to be concerned about the entrapment of those peptides in the vesicles, right?
Yeah, in the endosomes, yes, absolutely. And you can't predict that either. So you have to find it out.
Would you allow me to suggest using crotamine? Crotamine is quite good. It can release.
Crotamine is a bit too long.
Yeah.
The shorter peptides are more interesting.
Yeah, that's true. But now we have the synthetic analog, so it's still a little bit expensive, but it works quite well.
You know, every group has their own peptide because there are so many peptides. And it's very difficult to convince somebody to work with your peptide. Extremely difficult.
That's true. You're right. So I do have another question from Wagner. So he's asking if you can use CPPs for exosomes.
Yes. I said yes.
Okay, thank you. Patrice is also saying thank you for you. And Professor Luis Juliano Neto, he's asking if you can say something about the development of specific cell binding cancer cells.
Specific cell binding cancer cells?
Yes.
I don't understand the question.
I think that he's asking about specific CPPs with affinity for cancer cells.
Ah, okay. I mentioned already that there is a huge amount of homing peptides and cancer homing peptides as well, which have been screened in phage display library, which are actually screened in vivo in tumor model mice, for example, and there are quite good ones available. Some of them are in clinical trials. But there are also other technologies. Homing peptides are well developed, but there are other ligands you can use for cancer cell. Any cancer cell surface receptor could be used as a target, probably. But homing peptides are better because they are screened in vivo for cancer model, and sometimes the receptors are not even known when you find them. When you have to patent it or make a drug, you have to find out what kind of receptor it interacts with, and this shows sometimes pretty difficult. But some of them are known also, so people are using mostly these.
Yeah, that's true. By the way, do you have an idea what is the physicochemical features that determine if the peptide is going to be internalized by endocytosis or it's just crossing?
No.
It's very unpredictable, right?
I don't know. This is what I would like to predict, but I think nobody asked this question.
Yeah, me too. Neither. And it does not seem to be only the size, right? It's not only the charge, not only the size. So there is something else that I cannot understand.
Well, I showed you the zeta scales, and there are 5 of them in this simplified system. So it's a very complicated procedure of cellular uptake. You saw the scheme, the kinetic scheme. There are many steps: interaction with a receptor, interaction with a plasma membrane, simultaneously interaction to get through the or other side to a cell, and more kinetic steps. And to predict that, to quantify this prediction is very difficult. So usually you do this prediction without knowing these steps, or not using knowing of these steps. Well, why don't you solve it?
Yeah, yeah. that's a difficult. Yeah, it's a challenging.
Yeah,
Doctor.
but ask this question and then just use some prediction and take a trainer set and then go on. The question is that usually these two procedures are in parallel, and if you are not able to find a situation where only one of them is clearly the component, I think.
Yeah, you're right. The point is, I'm always thinking why Futaki's sequence didn't work. I mean,
Yeah.
They have the chart, they have the size, but it seems that they are missing hydrophobic groups or big size groups.
Well,
So
and this depends on the cargo also. Cargo will change the properties immediately.
yeah.
So if you want to know the properties, you have to study the CPP cargo conjugate instead.
Yeah, but somehow it works for proteins, right? I mean, those proteins have already the right CPPs, just carrying them. So they
Yeah, usually people are using TAT for protein delivery, and some people say that it works, and the amounts which TAT takes in are small. So maybe in small quantities, protein delivery with TAT is okay, but we have seen that many other CPPs do the same. But it's again, many people tested TAT and it worked, so nobody wants to use anything else.
Yeah, that's true. I have a last comment from Professor Luis Juliano. He says that specific cell binding peptides or CPPs are important for theranostic PET-CT assays or diagnosis. And so if you allow me, I would add that actually crotamine is very specific, and we are doing some work on PET-CT with high success.
Yeah, that's very good.
The point is that it's too big and too expensive, as you mentioned, but it works.
I know.
The point is how to minimize this structure. But Juliano, if you are interested, we can discuss more about this. We have quite good data on that.
Did you try to make it shorter?
Yeah, I tried. It doesn't work. It's just changed the way that it is internalized. So usually crotamine is internalized by endocytosis, even if with or without cargo. But if you reduce the size, it just goes through the membrane and lose the specificity.
But that's what you want.
Yeah, but if you lose the specificity, then you're lost.
Well, you cannot have it all.
That's the point.
Choose something else. Make crotamine shorter if you can, so it acts as a CPP, and instead of what you took away, use the short homing peptide instead.
Yeah, actually, I have a hypothesis that maybe you agree or not, but crotamine is 42 amino acid residues, and there is a three-dimensional structure, and there is a charge all surface in different surface. So I think that the surface determine the specificity for proliferating cell is not the same surface that is binding to the cargo. Okay. So when I reduce, then I might have just one motif and not the important motif. I don't know which one is. That's the point. So it's not only charge. It's charge and something else that I'm missing.
It's always charge and everything else.
Yeah.
Hydrophobicity component is quite important.
Yeah. This is why it's difficult to reduce. When you start to cut off, you start to lose some of the three-dimensional distribution. You cannot do that with a linear peptide.
Yes, I would choose another peptide. If it's too long and then strange sometimes, and you know there are so many other possibilities. So I can't I can't say.
Okay, so if there is no more questions, I would like to thank you, Dr. Langel, for your time, for your wonderful presentation. It was really amazing. We learned a lot with you, with no doubt. I'm very anxious to see your next book, the release of next book. I studied very detailed, and I hope we can just continue our discussion and maybe do some collaboration in the near future.
Thank you. I'm very happy to be in Brazil and hear the birds singing.
Yeah,
So,
please. Come
until
over.
the next time.
Yeah, no
Thank
problem.
you. Bye-bye.
Okay. Thank you. Bye-bye.